this is just the last piece. It doesn't affect really the way that you make Punnett squares or predict crosses, but it's giving you a bit of information about how stuff works in real life. We got a bit of a glimpse of how genes can affect each other in the last example about epistasis. That was the one with the rooster combs, and it would be similar to the cat fur color example, number 8 out of the homework for lesson 8. But there's more than just genes that can affect genes. The environment can affect the expression of genes. So if you'll recall from our last unit, genes being expressed means that they get converted into a protein. DNA makes messenger mRNA. Messenger mRNA goes to the ribosome and a protein gets produced. That's the gene being expressed. There are environmental factors that could, they don't always in the case of every gene, but that could affect whether or not a gene is actually expressed. Things like diet, temperature, oxygen level, humidity, light cycles. So how long there is day and how long there is night, which can vary over the years, so seasons. The presence of mutagens. All of these are examples of things that can impact which of an animal's genes are expressed, which ultimately affects the animal's phenotype. So just as an example, this is a real thing that really happens. There's a species of freshwater turtles who have thermosensitivity. What happens is that the egg will be laid. In the beginning, the gonads, which are the reproductive structures, are indifferent. That means they're not male or female. They haven't decided what they're going to be yet. Between about the 28th and the 52nd day of the egg developing with an embryo inside of it, the embryo is thermosensitive. If the temperature is higher, then the thermosensitive gene expresses as female. If the temperature is lower, it expresses as male. And so the temperature in which the eggs are placed will determine whether or not the SOX9 gene is expressed or repressed. If it's expressed, male. If it's repressed, female. Now, you don't have to know the details of this particular example. It's just to show you that just having a gene doesn't necessarily mean that it's going to be expressed. You could have a gene, but because of an environmental condition, that gene never gets turned on, never gets activated. And so it's really important to remember that in real life, even though a person might have a gene, they might never express it. We're going to come back to genes that affect especially the sex of the offspring. There are several different scenarios in which in humans, genes can be activated or repressed or mutated, and a person could have a gene that's supposed to make them male, but it never gets expressed properly, and the person ends up having female structures even though their genetics say they should be male. It is a very cool thing to research. Now, like I said, we'll come back to that at the end of the course when we talk about human reproduction. But if you were like, wow, that's interesting, I didn't know that was possible, you could certainly be like, oh, I'm going to do my de facto about that and look some stuff up, because we'll definitely talk about it later on. Now, that's the last little piece of this lesson on multiple alleles. This would be where the content for the quest tomorrow ends. Same format as the last one, 4 questions, 2 multiple choice, 2 numeric response. But I wanted to wait till tomorrow so we could make sure we finish this. Does anyone want to ask anything? So that takes us in to the last lesson in this unit. We're going to take a look specifically at how we map human genetics. And we use these things called pedigrees. And I put a fun little cartoon there of a caveman drawing cave people on the wall of the cave. But a pedigree is a fancy word for a family tree. And I bet you've seen or heard of a family tree before, even if it's not in the scientific context. This is where you might recall I said, hey, I'm going to ask you to make a family tree, either for your own family or for a fictitious family that I will tell you about. So this would be where, if you know whether people in your family can roll their tongue or what their earlobes look like, it's going to become very useful. Now there are some symbols that we will use when we draw family trees. All of them can be found on your data sheet. So if you put that data sheet away after we were done looking at mRNA codons, you'll want to locate it because it has these symbols on it. If you lost your data sheet, the file, the electronic file for the data sheet is posted in Google Classroom, and you can always access it there or print yourself a new one. And I always bring data sheets to tests with me, just in case that was ever a worry that you have. So you don't need to memorize these, but you do need to know that they're there. We'll always represent males with squares and females with circles. Anytime people are mating, so they're going to have kids, we put a horizontal line between them. If the mating is between close relatives, which won't happen a ton, but you might see in some pedigrees, you would see a double line. This would be in the scenario where perhaps cousins marry each other, and you might think, that's kind of odd. Does that really happen? Probably not so much here and now, but in the past and in other places, this is still something that happens with enough frequency that we need to know the symbol for it. Mating between close relatives was actually fairly common and still kind of is in royal families. Maybe it's getting a little better where royal people are allowed to marry non-royal people, but a few hundred years ago, if you were someone who was royal, really your only option was to marry someone else who was also royal, and there's only so many royal people in the world, and so chances are you're kind of related to each other. I had been watching The Crown this summer on Netflix, and I didn't realize quite how closely related Queen Elizabeth and her husband Prince Philip really were. Because even though they have different last names, they're actually related to each other through Queen Victoria from a few generations before that. And so they have relatives in common. And so if the mating is between close relatives, you'd see a double line. I don't expect you to know how to predict how close people have to be to use this symbol, but if you see it, that's what it means. Now the next one kind of shows you how we number things. When we're looking at a family tree, each row or level represents a generation. The first one will always have the Roman numeral number 1. Their kids and anybody who's in that age group of generation will always have a number 2 Roman numeral, and we could go 3, maybe even 4. Most pedigrees that you'll see won't have more than 4 generations because it gets just too big to draw. Within each group of offspring, we number we place the children oldest to youngest. We try to do so in the whole generation, but often it's impossible. So for example, these first 2 people have had 3 kids. We could put them in age order, oldest on the left. That's no problem. But once they start having kids, unless they have their kids in sequence, where the oldest one has all of his kids first, and then the second oldest one has kids, it's impossible. So as long as you have each family grouping in age order, you have done it correctly. We'll always use Arabic numbers, which are just regular numbers, to number people in a generation starting at the left. That means that every person in a generation should have a unique number. So for example, this person here would be person Roman numeral 1-1. So I'm going to use a Roman numeral and an Arabic numeral to represent a person so you know exactly who I'm talking about, even if you don't know people's names. Now, if there happen to be twins, there are 2 different versions of the twin symbol. Children who are identical twins will be represented with almost sort of a triangle. Notice over here in this family grouping, these 3 kids aren't twins. So they each get their own separate vertical line coming down from their parents. When there are identical twins or non-identical twins, see how the line is diagonal instead of vertical? And if they're identical, there's another line connecting them at the bottom, just so that you can tell that that's the type of twins that they are. The last thing you need to know is what colored in versus not colored in means. Now, every pedigree will be different because we're looking at a different characteristic all the time. But if an individual is colored in, it means they are affected. In other words, they have whatever characteristic we are talking about. Now, each individual pedigree will tell you, this is a pedigree for blue eyes, or this is a pedigree for a hitchhiker's thumb. So the title of the pedigree will tell you what it's for. The thing that's colored in might be a dominant trait, might be a recessive trait. There's no rule there. It just means that the person has whatever trait it is we are studying. If the person's symbol is not colored in, it means they do not have the trait that we are studying. Now, we'll often make these pedigrees for diseases, and so that's why the word affected is there. They're affected by this. But we could make pedigrees for non-disease characteristics as well. Now, does anyone want to ask anything about the symbols? You can always look them up right there on your data sheet. So what we're going to do now is look at different patterns of inheritance and what they would look like in pedigree format. So if you didn't take a copy of those blank pedigrees, there are some on the table up here. You probably want one, because I don't know about you, but I don't want to draw this big family tree 5 times today. So I'm going to use this same pedigree and show you what different patterns of inheritance would look like. The first pattern of inheritance is autosomal dominant. That means we're looking at a dominant allele that you would find on an autosome, not a sex chromosome. The 2 things in the title are some examples of characteristics that you would get because of a dominant allele on an autosome. The first is polydactyly. Anyone know what that is? Poly means many. These are your dactyls. So polydactyly means having more digits than you should. Often it means a 6th finger on each hand. It doesn't have to be both hands, and it could be more than that. But that's polydactyly. Another one is Huntington's disease. That one maybe a little bit more serious and severe than just having like a cool extra finger. But those are some examples of traits that are caused by an autosomal allele on or an autosomal chromosome with a dominant allele. Now, if you look at the pedigree that's here, there are 3 generations, and the people are numbered in each generation. So that part is already done for you on those handouts. What I added in first was whose symbol got shaded in. I colored in grandpa up here, his daughter and his son, the daughter's daughter, and the son's 2 kids. And a lot of the time, that's what you'll have. You'll have a pedigree with some symbols shaded in and other ones not shaded in, and that's all that you know so far. And so that would be our starting point. Now, figuring out their genotypes is something I did once I decided that this was autosomal dominant. So here are some clues, things that you can look for on a picture of a pedigree that will tell you, this is autosomal dominant. The first thing that you will see is that if the condition is autosomal dominant, it shows up in every generation. You see a vertical pattern. Affected parents have affected kids. Affected kids have affected parents. You see that it's getting passed on from one generation to the next. The second thing that you'll see, because it's autosomal, because it's not linked to a sex chromosome, is that males and females will have the condition with equal frequency and severity. That means you should expect to see about the same number of women, about the same number of men with the condition, and that it should affect them approximately the same way. Now, especially for this point, you have to understand that in real life, there aren't always equal numbers of males and females in a family. And so if I was looking, and I look at this pedigree, it just so happens that there are 1, 2, 3 affected females, 1, 2, 3 affected males. That's exactly equal, so you're like, oh yeah, that's exactly equal, so I've filled in condition number 2. Sometimes though, you might maybe see 4 males and 3 females, That's pretty close to equal. It's not perfect. And so especially with number 2, don't expect for it to be exactly the same number, but expect for it to be close. Now the third point, I find a very useful one. Unaffected individuals do not have children with the condition. That one is super important. If you were unaffected, that would mean that you do not have this dominant allele. If you had it, it would show up because it's dominant. And so especially point number 3, I use to help me decide if I think the trait is autosomal dominant or autosomal recessive, which will be the next one. So if I take a look, there is no example in this pedigree where unaffected individuals have children who have the condition. Over here, person 2.5 and 2.6, neither of them is affected, and their child also isn't affected. It's not a ton of data, but I don't have any examples where 2 unaffected people end up having an affected child. Now 4 is giving you some percentages, and sometimes those are hard to see by looking at the pedigree, but each child of an affected individual has a 50-50 chance of being affected, regardless of sex or birth order. And that's because it's autosomal, and that's because it's dominant. Now, frequently, not 100% of the time, but frequently, individuals who have 2 copies of this allele for whatever disease it is have a more severe form of the disease. It's not a 100% rule, but it's very common. And so if you were to ever see this, that could be another piece of evidence that you could use to decide that it was autosomal dominant. Now I want you to know, right, these are clues. These are pieces of evidence. You might not have every clue all the time, but you certainly will have some of them. If I'm going to ask you to decide whether a trait is autosomal dominant or some other pattern of inheritance. Now, if I go back to my pedigree, I filled in what everybody's genotype is in this pedigree, and here's how I did so. I had decided that this was an autosomal dominant trait. Even though the title told that to me, that would have been my decision anyway, because it's a vertical pattern and males and females are affected with equal frequency and severity. I don't know about the 50% chance of being affected regardless of sex or birth order unless I start making some Punnett squares. I don't see unaffected people with children who have the condition, and I don't have any information about whether it's more severe, so I can't really use 0.5. So based on that, I'm going to say that everybody whose circle or square is not filled in is homozygous recessive. They don't have this condition that we're looking at. So the first thing I would have done is filled in for everybody with a white symbol that their genotype was little h, little h. I'm using H's here for Huntington's only because capital and lowercase p looked too similar and I didn't want to use them. Now for everybody whose circle or square is colored in, I know they had to have at least 1 copy of the capital H. At least 1. And you'll see that I underlined their second allele, everybody, because at an initial glance, I'm not 100% certain if they're homozygous or heterozygous. But you'll notice that I noted down here, either by looking at the offspring or the parents, I know everybody's genotype. For example, let's take Grandpa up here, person 1-1. He definitely had the allele, right, because he has the trait, he has the disease. But he has a kid, his 3rd child, their son over here, who's homozygous recessive. So I asked myself, where did this boy get his alleles from? Always 1 from mom, 1 from dad. So yes, mom was homozygous recessive. She must have passed one of those on, but dad had to as well. He couldn't have gotten 2 alleles from mom. That goes against the laws of segregation. He had to get 1 from each parent. So dad must have had a recessive allele, otherwise, how did his kid get it? Using that same logic, I could fill in person 2-1's genotype, And I could use her mom or her kids. If I look at her kids, I would say, she has a kid who's homozygous recessive. This kid has to have gotten their alleles from both of their parents. Dad clearly gave one, but mom had to give the other. For person 2-3, I would have to look not at his kids, but at his mom. His kids don't really give me the answer. His kids have the trait, but his mom was homozygous recessive. If your parent is homozygous for a gene, they have to pass it on to you. It's the only choice. For these kids down at the bottom, person 3, 3, 3, 4, and 3, 5, I'm going to use their parents to fill in their missing genotype. All of them have one parent who is homozygous recessive, and so I know they must be heterozygous. So that would have been my logic for filling in the genotypes for everybody on this pedigree. First, I had to decide that it was autosomal dominant. Then, I used the clues that I have to fill in genotypes. Now, this one worked out. Sometimes you might have question marks left at the end, which is okay. You might remember from the last little quest that one of the mom's genotypes was uncertain. I didn't have enough information to decide completely, and that might happen. This one didn't have it, but it could. Now, before I go to the next pattern of inheritance, does anyone want to ask anything about this one? So another possible pattern of inheritance is autosomal recessive. You'll notice I've got the same family in my family tree. A couple of examples of autosomal recessive disorders would be something like cystic fibrosis. Cystic fibrosis is a condition where lungs fill with fluid. It's not a great thing. Tay-Sachs is very often fatal. And so you might not even be able to make a family tree for some of these conditions because people don't live a super long time. I'm using T's really because capital and lowercase c looked too similar. Tay-Sachs is a neurodegenerative disorder. There was an episode of Law and Order once where the people had a kid with Tay-Sachs and then they had a second kid and they thought he had it too. So the parents euthanized their own child. Then they realized the kid didn't have it and they killed what would have been a perfectly healthy kid. It was very sad. But Tay-Sachs is a condition that we'll talk about actually in the next unit too, because it tends to show up more in certain populations than in others. Now, if a trait is autosomal recessive, that means it's on an autosome, not a sex chromosome, and that a recessive allele causes the particular trait. So we know from before that to have a recessive trait, you need 2 copies of the recessive allele. So that would be true for cystic fibrosis and for Tay-Sachs. Now here are some clues. Same as before, you might not have every clue all the time, but you should have at least a couple of them to help you decide that it's autosomal recessive. The first clue is that the condition will typically appear in one generation, but nothing in their parents or offspring. So that means, in other words, it skips a generation. The second thing that you'll see, which was the same point as we had on the first one, is males and females being equally affected. It might not mean exactly the same number of males and females, but anytime I make a test question with this on it, I make it either perfectly equal or very close to equal, okay? In this particular example, if you look at who I colored in, I colored 1 male and 1 female. So you definitely have the same number of males and females affected. The third thing talks about asymptomatic heterozygotes. These people are carriers. They don't have any symptoms of the disorder. You would never know that they had the allele because they have 1 copy of the change gene, of the gene that causes the disease, and 1 copy of, in this case, the dominant normal allele. That means that they don't actually show the disorder. If you have 2 carrier parents, so these 2 people would be carrier parents. They have a 1 in 4 chance of having an affected child. And sometimes the percentage might not match the data because, of course, they have 3 kids, not 4. But remember, it's just a prediction. Each time they have a kid, those are the chances. Same thing for number 5. If you are a full sibling of an affected individual, you have a pretty high chance of being a carrier, because it means that your parents must have had the allele to pass on. If you have offspring from an affected individual, they will definitely be a carrier, unless the other parent also passed on the allele, and the other parent is a carrier or is affected with the condition. But if you had an affected parent, at the very least, you're a carrier of the disorder, and you might even have the disorder if your other parent had the allele as well. Now, 4, 5, and 6 are sometimes hard to see because sometimes you don't know if people are homozygous or heterozygous. Sometimes you don't know what the other offspring are. But if I look at the first few clues, I definitely see that it skips generations. Grandma and grandpa are unaffected, but they have a kid who is affected. His children are not affected, but he has, over here, a niece who is affected. So it skips generations. It'll go from 2 unaffected parents to an unaffected child, or 2 unaffected parents to an affected child, like this. You'll also notice in a lot of cases that there are just fewer people that have the trait because it's recessive. Now, if you take a look, I started out with these 2 people who have the trait. They're homozygous recessive. And anybody who didn't have the condition, Tay-Sachs in this scenario, got at least 1 capital T. So everybody else on the Punnett square got a capital T. Now, for some of the individuals, I had enough information to determine for sure whether or not they were a carrier. For others, I didn't have enough information to decide for sure. There's a pretty good chance, but I can't be certain. So for example, grandpa and grandma. I decided for sure that they were carriers of that recessive allele because their second child was homozygous recessive. And just like in the last one, he had to have gotten those alleles from both of his parents. Each parent contributes 1 of the 2 alleles. Because person 2-3 was homozygous recessive, I also knew that their children were carriers. Okay, so I was certain. So this point, offspring of an affected individual will be a carrier. That I used here to fill in the genotypes for person 3-4 and 3-5. Over on the right-hand side, I used person 3-6, who was homozygous recessive, to determine that their parents must have been carriers. Parents didn't have the disorder, but they had an affected child, so they must have been carriers. That's the only way she could have the disorder. All of the other people on the pedigree have a question mark, and that's because I don't have enough information to decide for sure if they're a carrier. I know that these siblings of an affected individual have a good chance of being a carrier, but that doesn't tell me for sure. It just tells me that there's a good chance. So that's why person 2-1 has an uncertain genotype. Person 2-2 isn't a sibling, right? They come from somewhere else. That's just the person that she is having kids with. And so I don't know anything about his parents, so I can't be certain of his genotype. So it's okay to have uncertainties, and in fact, you likely will in scenarios like this where there's only a few affected individuals. Now that's autosomal recessive. Does anyone want to ask anything there?